Hello viewers, welcome to Physiology. In this video, our main focus is on the two phases of photosynthesis. Photosynthesis, as we all know, is one of the most significant processes. This process, which is done by the green plants, produces food not only for themselves, but also makes it available for all other organisms. Besides making food, it also releases free oxygen into the atmosphere, which is utilized for the process of breathing. So let's learn a little more about this process of photosynthesis, which comes under the two heads: a light dependent phase called the light reaction, where it starts with the activation of chlorophyll, followed by the photolysis and photophosphorylation. The end products released by the light reaction also helps to drive the biochemical reactions, which helps in the manufacture of glucose. So let's try to understand a little better the mechanism of the processes which are headed under the light reaction as well as the biosynthetic phase. Now, the thylakoid membrane contains specialized molecules that work together to perform the light reaction. Light is absorbed by the protein pigment complexes called the photosystems. Namely, there are two photosystems recognized in the plant, that is photosystem 1 and photosystem 2. These photosystems which help to transform light energy to chemical energy, and that is done by exciting and reshuffling the electrons in a chain-like fashion on the thylakoid membrane. This chain-driven reaction, which is called the electron transport chain. This starts with photons of light hitting chlorophyll, a light-absorbing system in photosystem 2, lifting the chlorophyll atom from its ground level to an excited level. The excited electrons are then passed on to an electron carrier. Meanwhile, the water splits and releases electrons. So these replace those electrons that were lost at photosystem 2. The byproduct formed here is oxygen, which is eventually released free in the air. The hydrogen ions, which are released into the thylakoid lumen, the excited electrons move now to the cytochrome complex. Some of the energy from the electrons is used by the cytochrome complex to transport additional protons to the lumen. The second electron carrier, a protein inside the lumen, receives the electron and passes them to photosystem 1. These electrons have now lost most of the energy that they gained from photosystem 2. Photons of light hit chlorophyll in photosystem 1 and excite the electrons again. These electrons are then replaced and passed on to the third electron carrier. Finally, these electrons are either recycled or they interact with an enzyme and NADP+, that is the final electron acceptor of the light reaction, to form NADPH. Some of the energy from light is now stored in the reduced NADPH form, and here this molecule, which can now release some energy from the transfer of electrons, that is established a proton gradient across the thylakoid membrane. Protons that are accumulated in the lumen diffuse into the stroma through an enzyme called ATP synthase. It uses the potential energy of the gradient to combine ADP with inorganic phosphate to form ATP. Thus, potential energy is transformed to chemical energy, which is stored as ATP. This ATP and NADPH now has stored energy from light reaction. This energy can further drive the Kelvin cycle or the biosynthetic phase, which can ultimately lead to the production of the end product of photosynthesis called glucose. Now let's see how does that biosynthetic phase operate in order to produce glucose and the outcome of the oxygen along with all the other end products of photosynthesis. The overall purpose of Kelvin cycle is to convert atmospheric carbon dioxide into glucose, which is required for the cellular activities or to build new structures. This site of Kelvin reaction is the stroma of the chloroplast. It uses the ATP and the NADPH for the conversion. ATP is adenosine triphosphate and NADP is nicotinamide adenine dinucleotide phosphate. And when it is with an H, it is reduced form of the compound. Now, the three phases in the biosynthetic process are carbon dioxide fixation, followed by carbon dioxide reduction, and finally the regeneration of RUBP, ribulose bisphosphate. Now, this happens with the starting of carbon dioxide, which is attached to RUBP. The RUBP is a 5-carbon molecule, whereas carbon is a single carbon molecule. The enzyme used here is Rubisco, that is the most abundant enzyme present on Earth. This results in the formation of an unstable 6-carbon molecule, which then breaks down into 2,3-PGA or 3-phosphoglycerate, or we may also call it 2 molecules of 3-phosphoglyceric acid. This 3 denotes the position of the phosphorus which is attached on the carbon atom. So it is attached on the 3rd carbon atom, so it is called the 3-PGA. The process of converting CO2 to glucose begins. In the second step, what we find is this is called the carbon dioxide reduction, because the electrons and the energy are added to carbon dioxide during this process. Now, a sequence of electrons uses the NADPH and some ATP from the light reaction. These 2 molecules supply the much needed electrons plus the energy for the reduction of CO2. Now, electrons are added from NADPH through a series of reactions. The 3-PGA or the 3-phosphoglyceric acid is reduced to form a carbohydrate which is designed and called as G3P. The adenosine diphosphate or the ADP and the NADP plus return to the thylakoid to be converted back to ATP and NADPH via the light reaction. Now, one of the G3P molecules is set aside for the formation of glucose, but majority of the G3P molecules move forward into a 3rd phase of Kelvin cycle. In this, the ATP is used to combine the rest of the G3P molecules to form ribulose biphosphate molecule. This ribulose bisphosphate can then combine with the additional carbon dioxide, continuing the carbon reactions. To form a glucose molecule, the cycle actually has to turn 6 times, because each turn of the cycle adds only one carbon atom from the incoming CO2. And since glucose is a 6 carbon compound, so until and unless the addition of CO2 occurs 6 different times, it would not lead to the production of the 6 carbon compound called glucose, which is the ultimate need for the plant by this process. Thank you for watching. Please do like, share and subscribe to my channel.